Hi everybody. Today we're going to be talking about genetic engineering, and I wanted you to read Jonathan Glover's article on this topic, and so that is where we will start. Okay, so Jonathan Glover, his article is called Questions About Some Uses of Genetic Engineering. Okay. Sorry about that. I forgot that you guys can see that whole screen even when it does that. Hold on, I'm going to pause because I need to get another document up top. Okay, so sorry about that little glitch again. Let me give you an outline of what I want to cover. First, we'll talk about context. Then we will get right into Glover's article. We will talk about nature versus nurture, eugenics a little bit, negative genetic engineering, and positive genetic engineering. And within positive genetic engineering, I'm not going to put them all here, but we are going to talk about 4 objections and replies. Okay? So that's an outline, what we're going to talk about. Okay, let me start with a little context first. So first, what is genetic engineering? Okay, so genetic engineering is sometimes also called genetic modification, is the direct manipulation of the genome using using biotechnology. Okay? There's a contrast using selective breeding. Okay? When you breed selectively, only let some people have babies, prohibit other people from having babies, or doesn't even have to be people. It could be dogs, it could be cows, it could be corn. Okay? If you use selective breeding, then that's not genetic engineering, that's called eugenics. Okay? Using selective breeding, I'll even emphasize, for the same goal is eugenics, which is different. You know, it can have the same goal as genetic engineering, but it's a different technique, selective breeding. Okay, an organism that has gone through genetic modification is called, some of you may know this already, like you can see these things from your grocery stores, you know, like especially those Whole Foods type organic grocery stores. These are called GMOs, or for genetically modified organisms. Okay? And that actually leads to the next topic, which is GMOs are everywhere. Okay? We first genetically modified bacteria, including to make insulin. We just turn bacteria into our slaves and have them churn out, like just put genes into these bacterial cells to basically turn them into factories to make insulin for us, and then we collect the insulin. And when was this? About 1982, not that long ago. And genetically modified food, like corn and so on, has been sold since 1994. All right? Lots of crops these days are genetically modified in some way or another to make them hardier, to make their fruits more plump, to make them, more resistant to bacteria, to make them grow more quickly, etc., etc. Okay? So possible uses include, as I've just said, agriculture. Okay? Crops like resistance to pests. Okay? Cows to put more protein in their milk. Okay? The scientific consensus here is that this is incredibly safe. It is no more dangerous than breeding crops to have the resistance, or breeding cows so that you only let the cows who produce the most protein in their milk have babies and have more cows. The scientific consensus is the genetically modified organism is safe. But this is not our topic today. Okay. Another thing it's used for, as I said a little bit earlier, making drugs. For example, insulin, and hopefully sooner rather than later when we get a vaccine for COVID-19, making vaccines. We turn bacteria into our little, like I said, our little slaves. We put certain snippets of DNA into these bacterial cells, and that instruct the bacterial cell, here's your job for the rest of your life. Churn out the vaccine to COVID-19 or whatever it is. Okay, and that's what they do, and then we just call them other hormones that we might want to make. We also can have GMOs for animal models, such as genetically modified mice. You have certain what are called knockout mice, where you can genetically modify the mice to knock out a certain gene, and then you can see what happens. And this can be useful, both just to see what happens when you knock out certain genes, and also if you want to use those mice to test certain hypotheses you might have about this or that theory. Okay. Now, our focus is not going to be any of these kinds of uses: agriculture, for crops, cows, mice, bacteria. Our focus is going to be people. Is it okay to genetically modify people? Okay, let me give you some distinctions also, so that we are kind of on the same page and we understand at least a little bit of science and the different kind of issues that are, issues at stake. So some distinctions. Okay. So one is a distinction between negative genetic engineering versus positive genetic engineering. And this is a distinction that Glover makes right away. And it's one that in the second article I'm going to ask you to read for Thursday, that author Michael Sandel also just kind of presupposes that we know what this distinction is. Okay? In negative genetic engineering, you replace faulty genes with normal copies. So basically, you bring diseased people back up to normal. Okay? And sometimes this is called, aka, gene therapy, because the idea is it's a therapy using genetic modification to get you back up to normal. Okay? Positive genetic engineering, though, sometimes this is just called genetic engineering. Without this positive phrase, it's a little bit like if you've listened and watched my video on the definition of death, it's a little bit like how sometimes people just call whole brain death just brain death. So similarly here, some people just use the phrase genetic engineering to describe what we would call positive genetic engineering. And what that means is enhancement. Okay? Bringing people above normal. For example, increasing intelligence, or height, or strength, or kind of like a focusing ability above normal, above what it would be normal for that person or maybe for the species. Okay? And this actually, the positive genetic engineering, is going to be our main topic. Glover does talk a little bit about negative genetic engineering. Sandel, the author I'm asking you to read for Thursday, also talks about it a little bit. But our main topic is not this, because most people think negative genetic engineering is totally fine, it's uncontroversial. The controversial one is the positive stuff, and so that's what we are going to talk about. Okay, another distinction. Somatic cell versus germline cell, or maybe sometimes this is just called germ cell. Okay, somatic cell genetic engineering, okay, is maybe it's better to describe germ cell first, because then somatic cell is just anything that's not germ cell. So germ cell genetic engineering is changes the cells that are engineered, are sperm and egg, so that it will change every offspring of that person who is created from that sperm and egg. Okay? Somatic cell genetic engineering is cells that are engineered are any cell other than sperm, egg. Okay, so that the changes kind of die with that person who was engineered. Okay, does this make sense? So if you change, for example, my liver, okay, that would be somatic cell engineering. It's not sperm, it's not egg, it's my liver. And my liver can maybe do all kinds of crazy things after the genetic engineering, but when I have kids, my kids' livers will not have the changes that my liver had. And so whatever changes we made to my liver will die with me. That's somatic cell genetic engineering. Now, in contrast, if you were to change, say, my sperm so that any kid born from my sperm has some kind of weird, freaky liver, okay, that means that any of that person's kids would also have a weird, freaky liver, okay? So germ cell genetic engineering, as you can see, the stakes are higher. More people are affected. Every one of your descendants will be affected by germ cell genetic engineering on you, whereas somatic cell genetic engineering on you only affects you, doesn't affect everyone else in the future, every one of your descendants. Okay. All right. Let me also briefly tell you about CRISPR Cas9. CRISPR Cas9. So this set of letters and acronyms, this alphabet soup, is basically, some of you who are biology majors may already know about this. This is CRISPR Cas9 is a relatively new technique, relatively new, kind of discovered and kind of streamlined only within the last 5 years or so, okay, for gene editing, not game, gene editing. It's very simple, precise, and fast, okay? So it makes the question of genetic engineering much more of a live question rather than just, theoretical science fiction. Okay? So if this were a biology class, I would go into some of the details of how CRISPR-Cas9 works. Okay? It's actually kind of cool, the science behind it, but this is not that biology class. I want to keep these videos as short as possible to not bore you with unnecessary detail like I sometimes did in my lectures. I apologize. So I don't want to talk about the biology of CRISPR-Cas9. Just keep in mind, this is a technique that's hot right now, okay? In the scientific community, this is a big deal right now. And it's led to lots of innovations, lots of different ways that we have already edited genes in people using CRISPR-Cas9 just in the last few years. It's kind of cool, okay? So among other things, using CRISPR, things we're already doing now. And this is some of the stuff we're doing now, as I say, has just started to become available in the last 5 years or so. So in terms of kind of biotech research, this is a very, very hot area. So let's see. We already do somatic cell gene therapy, okay? Mostly, this has been by taking bone marrow cells out of a patient's body, genetically modifying them, in test tubes, right, outside the patient's body, and then putting them back in. Okay? So one of the things we do, like with this, is, for example, cancer therapy. Okay? You can make, bone marrow cells, if you remember, I think I said this in class once, bone marrow cells make one of the most important things your bone marrow does is make your blood cells. For example, your red blood cells, your white blood cells, all of these are made in your bone marrow. So if you have a particular kind of cancer that is a cancer of the blood cells, like lymphoma, leukemia, you can get gene therapy for that. And that's really cool. So some people are already getting that. Another example of this that I just heard an interview about this maybe a month or two ago on NPR about sickle cell disease. They interviewed a woman who they were taking bone marrow cells out of her and editing the genes for that bone marrow and then putting it back in for those cells and then putting those cells back into her own body. And then those cells would produce new red blood cells. So if you know sickle cell disease, you know that basically the problem is the red blood cells that are produced in sickle cell disease don't have the usual rounded shape. Instead, they look like a sickle, like a crescent moon. And that leads to all kinds of problems. That kind of physical abnormality leads to all kinds of problems in the body. It's a very painful disease, kind of chronic, coming and going. It's a really nasty disease. Okay, so they had this woman who they took bone marrow out of. They modified the genes in these bone marrow cells, put the bone marrow cells back in her body, and then those bone marrow cells started producing perfectly round red blood cells. So she was essentially cured of her sickle cell disease, you know? And someone even asked her from NPR, they said, you know what this makes you, right? This makes you a GMO. You, lady, are a genetically modified organism. How does that make you feel? And she said basically, great, like this is curing me. I love the fact that I'm a GMO. I get to be cured of my sickle cell disease. Okay? Let's see. What else? Other diseases, so severe combined immune deficiency syndrome, immune deficiency, or severe combined immune deficiency. This is, I think I have not mentioned this in your class. I think I mentioned it in my other class. Like there's this, this phrase or this thing you may know of from pop culture called the bubble boy. There was one of these, I think, alive in the 80s. He had this disease, severe combined immune deficiency. What this means is he basically had no way to fight off any infection in his body. So if he gets a cut, he'll die. Like if he gets any bacteria like that goes through his skin or into his blood cell, he'll die because his body cannot fight it off. So what they did for him, and they do for all these kids who have this disease, they basically have him live literally in a bubble, like in a plastic bubble in your house or something, where he can literally never leave that bubble for his whole life. As soon as he steps outside and like breathes in, bacterial-filled air, he will die. Okay, tough life, right? So what they can do is take out, this is a problem, again, of the bone marrow not producing the correct white blood cells. The white blood cells, their main function is to help fight off infections. So they'll take out the bone marrow from these kids, replace the genes, put it back in, he gets a brand new life. All right. And just recently, I think I saw a report from March 4 of this year. Okay, they've been experimenting in vivo. In vivo means in the body, instead of taking cells out of the body, manipulating them and then putting them back in the body, they would directly manipulate cells in the body, hoping to change what happens. So now they're experimenting in vivo for certain kinds of blindness, where they are trying to manipulate or change the genes in certain cells right underneath the retina or something. So they're doing these experiments to cure blindness, and they're not taking cells out of the eye and putting them back in. They're directly going into these cells underneath the eye and manipulating the genes in there. It's all pretty cool. And this seems like, and most people agree, like this is all good stuff that we're doing. Potentially adding a tool to our little kit, our toolbox of how we fight cancer. Potentially curing sickle cell disease, that's fantastic, right? Bubble boys, maybe that'll be a thing of the past. And, blindness, who wouldn't want to cure blindness? Okay, let's see, what else do I want to say for context? Oh, okay, let me talk a little bit about positive genetic engineering, though, or at least legal regulations or legal status of positive genetic engineering. Okay? Because what I talked about, remember in the previous slide, things we're already doing now, these are all somatic cell gene therapy, aka maybe I should have said negative genetic engineering, right? These are all examples of curing some deficit, of bringing you up to normal from something below normal, okay? What about positive genetic engineering? What about using the same techniques to give people, to make people smarter, stronger, more focused, and so on. Okay, well, it turns out there is, as far as I can tell, no explicit regulation against this in the US, though many scientists and bioethicists are pushing for some. They're pushing the federal government to step in and say this should be outlawed. Genetically modifying people to make them stronger than normal, faster than normal, smarter than normal, etc. Okay? Probably, though, experiments testing this would never get out of the proposal phase, because all experiments funded by the government, which means basically all of them for biotech, because biotech tends to be very expensive. Well, I shouldn't say all for biotech, because there's like pharmaceutical companies and so on, but basically all of them for biotech in this area, okay, typically need some kind of federal funding, okay? All experiments funded by the government have to get approval by committees at the institutions where they are conducted. Okay, so if you want to conduct a study at Rutgers, there's a special committee at Rutgers. These are called IRBs, okay, institutional review boards, aka IRBs. You may have heard that acronym before, where they're conducted. So Rutgers has an IRB, Harvard has an IRB, every place that does research has an IRB. They need to approve all of these experiments, and my sense is no IRB would ever approve, at least given the current climate about, or the current attitude about positive genetic engineering, I would be shocked if any IRB ever approved an experiment on positive genetic engineering, okay? But again, it's not that there's an explicit regulation against it in the United States. There's not, okay? It's just that IRBs will probably never approve this kind of research. Okay, now let's talk about Glover's article. Okay, so the first point he wants to make is this point about nature versus nurture. He says, he wants to make this one. He says, look, some people think that debating the ethics of positive genetic engineering is pointless because all the variation we see in humanity is due to the environment, and genes contribute nothing. Okay? If genes contribute nothing, then what's the point of modifying our genes? You wouldn't get anything out of it, right? So if genes contribute nothing, everything is due to the environment. The environment is nature. The environment is nurture in this case, and the genes is nature, okay? If genes contribute nothing, everything is due to the environment, then there's no point in even debating whether we should allow changing people's genes, because again, changing genes does nothing. Okay, Glover's response to this, okay, before I give Glover's response, one possible response is to say, wrong, genes do contribute a lot to how we are. It's not just the environment we're raised in, our genes do contribute a lot. Okay, there's some evidence about this, about a lot of things like intelligence and like success, that there's a lot, there's a big genetic component in these kinds of in those traits. Okay, but that's not Glover's response. What he says is, I should call him by his initials, Jonathan Glover, JG. Okay, Glover says even if all the changes or all the variety, I think he puts it variation we see is due to the environment, maybe all that means is that our genes are all very similar. So still, if we change genes a lot, we can make big changes in our traits. Okay? So for example, maybe all of our, for example, this is Eric's example, not something that Glover said. We all have 2 eyes, not 3. And this is something that is genetic. all of our genes for how many eyes we have are all basically the same, okay? But that doesn't mean that, if we ever got the science good enough, this is not practical. This is a very much a science fiction example. It will never happen in the near future. But hypothetically, if we knew enough about our own genome, we could manipulate the genome and give ourselves a third eye. Wouldn't that be cool? Okay? All right, so Glover, his basic point is this whole debate about nature versus nurture is irrelevant to this other debate about whether we should allow positive genetic engineering. The two debates are just kind of independent of each other. Okay, that's his point about that. Let's talk a little bit about eugenics also. Okay, so eugenics, remember, well, I guess I'll put it this way. There are three ways to change a population's genetic makeup. Okay, one is environmental changes. Environmental changes. For example, medical discovery, okay, poverty relief, agriculture, okay, all of these things plus more can change the genetic makeup of a population. So for example, just think about like the medical discovery of insulin. Insulin made it so that type 1 diabetics could live. And before it was discovered, they would just die. And often some of these type 1 diabetics would die when they were like pre-teenagers or like in their early teenage years before they had their own children. Okay? Type 1 diabetes has a genetic component. If your parents have it, or if a couple has it, then their offspring are at higher risk of having it than the offspring of people who don't have diabetes. So the medical discovery of insulin meant that we now have more people with type 1 diabetes than we did before. Because in the olden days, they would die. These days, they don't. Okay? So environmental change. Another way to change the genetic makeup is what I've already called eugenics. Okay? And remember, this is selective breeding. Now, eugenics, and then the third, I'll come back to eugenics in a second, but the third one is what is going to be the eventual focus, genetic engineering. Okay? So, and remember, genetic engineering is directly manipulating the genome using biotechnology, right? Get into those cells, change the genes in the cells, okay? Whereas selective breeding is decide who has babies and who doesn't have babies, okay? Now, eugenics has a really bad reputation. In fact, the word, if you have heard the word before, is associated with Nazis and like the Aryan supreme race and all these kinds of ideas. The Nazi ideology that only Aryans, like the blonde-haired, blue-eyed, white people, should be allowed to reproduce, and everyone else, if you have darker skin or darker hair color, should not be allowed to reproduce. Okay, because of association with Nazis and kind of forced breeding programs. Okay. Or forced sterilization. But, it's not just the Nazis. There was a eugenics movement that kind of was global, even before World War II, and including in the United States. There were people in this eugenics movement in the United States in the early part of the 20th century who said things like, imbeciles should just be automatically sterilized. Imbeciles being people who had a technical IQ below a certain level, like below 70% of on the standard deviation curve. And there were people who just thought, as a federal policy, we should never let imbeciles reproduce. That's a eugenics policy. Okay, very unpopular today, obviously. Maybe not obviously, but it is very unpopular today. Okay. Here's the reason that Glover wants to talk about eugenics is he wants to make a point about negative genetic engineering. Negative genetic engineering. Okay, so remember, negative genetic engineering was directly manipulating the genome to cure disease or remove some deficit, basically to bring you up to normal. Bring, not you, the patient up to normal. Okay? And Glover says there are certain conditions where we already accept what is technically a eugenics, I guess, proposal. And in those cases, we should also be willing to accept negative genetic engineering instead. Okay? So one of the examples of this, he says, is this. Screening followed by selective abortion for conditions like, Down syndrome, I think it's Down syndrome, Huntington's disease, spina bifida, okay? So the idea is you screen for these things in utero during pregnancy. If you discover them, many people think it's okay to abort that fetus that has Down syndrome or Huntington's disease or spina bifida. Okay, so it's okay to kill that fetus. So what Glover says is, if it's okay to screen for these and kill the fetus, surely it's better to screen and cure the fetus instead. He calls this, he calls it pure gain. Okay? Because the idea is we already allow screening followed by selective abortion. This is eugenics. Right? Deciding who gets to breed and what babies get to live because of their genetic conditions. Okay? And he says, hey, we're okay with this. But if that's okay, wouldn't it be better if instead of killing a fetus with Down syndrome, we just cure it? Or killing the fetus with Huntington's, we cure it, et cetera, et cetera. Okay, some of these are more technologically likely than others. So Huntington's disease, I think, would be pretty easy because it's a single base pair that's messed up. Spina bifida is like you don't have enough folate, and I don't know if you could actually use genetic engineering to cure that. Down syndrome, you have an extra chromosome, right? So, I don't know if you could use genetic engineering to cure that too. Maybe the genetic engineering is just get rid of that chromosome. But okay, so the examples may be hypothetical, but the point, I think, is a good point. Okay, so he thinks negative genetic engineering is fine. And basically everyone in the world, or the consensus, the medical scientific consensus is this is fine too. And in fact, as I've said before, we are already proceeding and performing and doing negative genetic engineering now, okay? And people are like in love with it. We think like it's great, it's fantastic. Okay, now let's move on to positive genetic engineering. Okay, so Glover defends positive genetic engineering. He thinks we should be allowed to change our genomes to make ourselves smarter, faster, stronger, et cetera, et cetera, et cetera, than normal. He thinks that should be allowed, and he wants to defend that view. He defends it by considering 4 objections and trying to refute all of them. Considering 4 objections to to positive genetic engineering and trying to refute all of them. Okay? So these 4 objections, which we will go over in the next slides, are as follows. So the first one says something like, we are at an evolutionary peak. Any changes in our genome will be harmful. Okay, that's the first objection. The second objection is genetic engineering makes us too different from our family. It would make these weird new generation of kids who are way too smart, way too strong, way too whatever, okay? Another objection is there's always the possibility of disastrous mistakes. I'll say disastrous and irreversible mistakes. So like we just don't know what we're doing, okay? And then objection number 4 is it's wrong to play God. Okay, genetic engineering, that's like not the territory we should be in. Either there's like a literal God whose territory that is, not ours, or it's a different objection about like leaving the decision about genetic engineering to a small group of people. Either way, wrong to play God. Okay, so we're going to consider all four of these, and we're going to see what Glover has to say about each of them. Okay. So, first objection. Let's see, what did I call this before? Here's another way of putting it. Overall improvement, or I'll say this. I'll just call it objection number one. Okay, because that's a good way to title the slide. So objection number one goes like this. Overall improvement is unlikely because, we're at an evolutionary peak. Evolution has already, evolution by natural selection has already made a human species about as good as it can be given the environment that we are in. Okay? So if we are going to muck around with what evolution has already done, we are going to make things worse. Okay? Because evolution by natural selection has already made us pretty much perfect. All right? That's the objection. Okay? So Glover, he says, no, that's not true. Natural mutations are sometimes beneficial. Okay? Therefore, artificial ones can be too. Okay? He also says, further, our goal isn't necessarily gene propagation. Okay? So just because, natural selection has made us perfectly efficient, even if natural selection has made us perfectly efficient at passing on our genes, still, maybe that's not the only goal we should have. Okay? And then further, finally, what we call a gain and a loss depends on context. So he gives this example of malaria and sickle cell trait. Okay, so those of you who know something about sickle cell disease know that if you have 2 copies of the sickle cell gene, you have the disease. If you have only 1 copy of the sickle cell gene, you have the trait. If you have the trait, you don't have any symptoms, but you can pass that trait on to your offspring. All right. Turns out, some of you may know this already, having the trait is protective against malaria. Okay, so sickle cell trait tends to be common in areas where malaria is common. All right. So what Glover says is, well, suppose we wipe out malaria off the face of the earth. This is technically possible. We could wipe malaria off the face of the earth if we just got around to doing it and had a coordinated effort to do so. Okay? In that case, sickle cell trait would no longer be good for us. It would be good for us not to have the trait, because if we didn't have the trait, we wouldn't have the disease. Okay? So what counts as good or bad just depends on the environment we're in. And in an environment where there's a lot of malaria, sickle cell trait can be good. In an environment where there's no malaria, sickle cell trait is bad. Okay, so that's his response to objection number 1. Okay, objection number 2. Okay. This is number 2. Positive genetic engineering makes us too different from our parents for purposes of bonding. Okay, now I don't even think that this is really a very good objection at all, so I don't want to spend a lot of time on it. I think this, Glover could easily have just deleted this objection from the article, and the article would have been shorter, but I think it would have been tighter also. I think this is kind of a frivolous example. But he makes a couple of points about it. He says, this is an exaggeration. It's not as if, my kid will be just wildly, wildly different from me. Like, he'll have 8 legs like a spider, and he'll have, the head of, a falcon, and, no, it won't be like that, okay? He'll be still very similar to me, okay? So it's an exaggeration. And then second, he says, we educate kids even though it makes them different from us. And I'll even add, I won't even put this in the slide, but I'll just say this, we move our kids. So my parents immigrated to this country, to the United States, from East Asian countries, and the values that they had over there were just completely different from the values here. And so when I grew up, I thought I was just really different from my parents. I thought there was not just a generational gap, but also a cultural gap because they grew up in China and so on, and I didn't, and so I cherished American values, they cherished Chinese values, right? So you can see how, that is something that they did that made me very different from them and made me, honestly bond less with them. I got into a lot of fights with my parents about things that I wanted to live the American way of life. They wanted me to live the Chinese way of life. All right? But we think that's totally fine. We think it should be allowed, even though it should be allowed for parents to move their kids, to immigrate to different countries and different cultures, even if it makes those kids very different from their parents. Okay. Objection number 3. This is a more interesting one. Okay, this one says basically, there's risk of irreversible disaster. Okay, basically, we don't know what we're doing in manipulating the genome. We could be making huge mistakes, and maybe these mistakes would just continue to propagate through the population, and we could never get rid of these mistaken genes. Okay, in the future. All right. Well, Glover says, obviously have safeguards. Okay? Now, there's a reply, though, which uses what's sometimes called the precautionary principle. The precautionary principle says if there's any risk at all of irreversible disaster, don't do the risky thing. Don't look at the, don't put it this way, don't weigh the potential benefits versus the chance of disaster. Just as a rule, stop doing that thing. Okay? So some people apply this, applications like nuclear power. Certain kind of global climate engineering, right? Like some people say here are certain things that we should do across the entire planet, spray a bunch of things into the atmosphere to reduce global warming. Other people say, no, once you spray a bunch of stuff into the atmosphere, you're never going to get rid of it and it could cause all these problems you don't know. Nuclear power, there's a chance of meltdown. So people who like the precautionary principle for nuclear power say, look, there's a risk of meltdown, which would be terrible. And once you have a meltdown, for multiple generations, you're not getting, that's going to be terrible for multiple generations. Just don't do it. Don't look at the chance of a meltdown. Just know that there is some chance. Don't do it. All right? Glover says, Jonathan Glover says, this is too extreme. We should have a principle of caution, not precaution. The principle of caution would say basically, have safeguards, be careful, okay, but be open-minded. But be open-minded too, okay. The slide is getting a little small, so let me make this a little bit bigger. Okay, an example of this might be, so I think this is itself an interesting debate. The debate about the precautionary principle. One of my former graduate students when I was at Colorado wrote his dissertation on precautionary principles, different ways you might formulate it that are better or worse than other ways of formulating it. Okay, because the precautionary principle seems to run into very powerful objections. So for example, consider driving. So for me, personally, as an individual, here's an irreversible disaster that happens, my death. For me, an irreversible disaster for me is my death. And I know that every time I get into a car, my chance of dying on the road, my chance of dying goes up whenever I'm driving or, in someone's car that someone else is driving, okay? The precautionary principle, if you were to apply it to driving, would say if there's any risk at all of the irreversible disaster, aka my death, never do that thing. In other words, don't weigh the potential benefits versus the chance. Just as a rule, stop doing that thing. So the precautionary principle applied to driving would say never drive your car, never get into a car ever. And of course, that's absurd. No one thinks that that's plausible. Okay, so Glover thinks the precautionary principle is also not plausible here. We need to be careful, obviously, but we also should just, be open-minded and weigh the pros and the cons. Okay, objection number 4. This is also a very interesting one. Okay, and this one is positive genetic engineering is playing God. Goff, playing God. Number 4. Okay, there are a couple of versions of this. Version A is there's a literal God, and it's his domain to mess with our genes, not ours. Like we're encroaching on a literal God's turf. It's his turf, not ours. Okay? Version B says it's wrong or dangerous to put the decision about what genes to edit into the hands of a small group of people, like politicians or even, scientists. Okay? So they're saying, oh my God, it's playing God in the sense of like, we shouldn't let, Trump's administration decide what genes we get and what genes we don't get, So that's a second objection. All right, so I'm going to go to the next slide and have one slide for each of these. Okay? So the version A, kind of the literal God. Okay? So Glover, so one thing you could say here that he does not say, you could say, what if there's no God, right? If there's no God, then this version of the objection just collapses. Okay, but that's not what Glover says. What he says is, it's hard to say where God's turf ends and ours begins. Arguably, medicine plays God, right? In this sense, like, is it God's turf to determine who lives and who dies, who gets type 1 diabetes and who doesn't? Well, why are we not kind of encroaching on his turf when we invent insulin and cure people with diabetes and so on? Okay? So there's that. All right. More interesting for us, I think, is version B. Version B, a small group of people deciding who gets what traits. Okay? So here, Glover's response is, I think, kind of interesting. He says, we shouldn't leave these decisions up to any small group of people. Rather, people should be free to get whatever enhancements they want. And he envisions this, he calls this a genetic supermarket, okay, which is analogous to a regular supermarket. Okay, so if you think about like food, there's no bunch of bureaucrats or politicians who get to decide what we all eat on Mondays and what we all eat on Tuesdays and what we all no. So what we do is each person has his or her own tastes, and we go to the grocery store and we just buy whatever we want. And people who sell food, they have to respond to demand. Some things are more popular than others. There are more of those options available in the supermarket. All right. So Glover says genetic engineering should be basically like that. It should be an area where people just want what they want and they're willing to buy it, and there's going to be people who are willing to supply it, and you just have market forces determine, using supply and demand, who gets what. Okay, so there's no kind of global top-down decision-making from the president or something about what is available. No, it's just things that people want, that's what companies will produce, and things that people don't want, companies won't produce that. Okay? Glover also, he concedes that this should be lightly regulated. Okay? If people want things that are really dangerous or bad for them, the government can forbid that. Okay? This would be, he doesn't say it, but I'm going to make this point for him. He doesn't say it, I'm going to make this point for him, that this is analogous maybe to the Food and Drug Administration, okay, the FDA, where we have a real supermarket for food where it's just supply and demand determines what food is made. And it's lightly regulated in that the FDA says, well, you can't sell poisons, okay? You can't put toxins into your food. There's certain things you can't sell, but outside of these things like toxins, poisons, whatever, just whatever you want. Whatever people want, that's what we should sell. Okay. All right, and that's basically all that I had to say about Glover's defense of positive genetic engineering, okay? So come back next time and we'll talk about some criticisms from Michael Sandel. Thank you.